Assalamu alaikum wa rahmatullahi wa barakatuh. I'm Adam Alkhawli. I'm a second year medical student. In today's lecture, I'll be giving you guys RNA transcription 1 and 2. Hopefully, if you guys study from this lecture alone, you're set for the exam. There's no need to go back to the PowerPoint. But for you people who like to go between the past session and the PowerPoint, I went along the same rhythm that Dr. Jeda went on. So basically, I went slide by slide and I put everything in the same order as he did. So if you want to go alternate between the past session and the PowerPoint, everything is organized for you guys. Okay? So before we begin, we'll just watch a quick video in order to get a quick overview on what we'll be taking. But then we're going to be breaking each part of this video down into more details. Okay? We'll stop here. Okay? For you who want the video, it's right here. Okay, so we'll be talking about transcription in this lecture or in this part session. We will not be talking about translation. Okay, keep those two different things in mind. Transcription is different from translation. Now, I was really short on time, so I wasn't able to make the PowerPoint all pretty and stuff. Bear with me, okay? Bismillah. So, the DNA is a double-stranded helix made up of many nucleotides. We already know that, right? This is the structure of the DNA. Now, proteins are the link between genotype and phenotype. What is genotype? Genotype is basically the DNA sequence. It's basically all the nucleotides put together, the G, C, A, T. Okay? As for the phenotype, let me just draw. Okay, as for the phenotype refers to the physical properties. So is it large, is it small, stuff like that, okay? So genotype is for the genetic sequences or DNA sequences. The phenotype is for the physical properties, okay? Now, for the gene expression, the definition of gene expression is the process by which DNA directs protein synthesis, okay? So DNA, what is DNA? It is just a storage of nucleotides, okay? So I have C, G, A, T, A, T, A. It's just nucleotides behind each other. It has no definite function. However, in order for us to get a function out of DNA, we need to convert it first into RNA, and from the RNA, we will get a product, mostly proteins, that can actually perform a function, okay? So the product of the RNA transcription is a protein which has a function. Okay. Gene expression has two stages, transcription and translation. As we said in this lecture, we'll be talking mostly about transcription. Okay. Okay, so right here we have the DNA molecule. It's a double-stranded helix. Inside we have gene number 1, gene number 2, gene number 3. Between these genes, we have spacer DNA, which code for nothing. Basically, they're introns, and they have no function that we know of as of right now. So basically, we have something called introns and exons. Okay? So the introns, they code for nothing. We don't need them. Okay? For the exons, they're what we're going to be using in order to make an RNA. Okay? So an easy way to remember it, exons enter. So they enter the transcription of RNA. Okay? So first off, the first step of RNA transcription or DNA transcription. Sorry, RNA transcription. The first thing, we have a double-stranded DNA. We're going to need to make it single-stranded. Okay? So we have a double-stranded DNA. Did I draw this correctly? I'm not sure. Okay. We need to make it single-stranded. Okay. So we're going to separate them and we're going to take this strand. We're going to use this as a template strand to transcribe the RNA. So what will this one be? We're not going to use it, right? So it's going to be called anti-template. Okay. So first off, we have the DNA, which is a double helix form. We're going to separate it. We're going to make it a single strand. We're going to take one of these strands, which is this one right here, and we're going to use it to transcribe an RNA. The one that we use to transcribe the RNA is called the template strand. What are the other names? Non-coding, and antisense. In the exam, he might get any one of these three names. So remember, non-coding, template, or antisense strand is the strand that we use in order to transcribe an mRNA. What about the other strand, the strand that we did not use? It's going to be called the anti-template, coding, or sense strand. So here we said non-coding, the other one is coding. Anti-sense, the other one is sense. Template, the other one is anti-template. Okay? What's the difference between the two? Let me find the anti-template strand, the other template for this one right here, okay? The other strand of DNA. So we have A, C, C, so it's basically T, G, G, T, 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 G, G, C, okay? I stopped right here. So this is the other strand of DNA from the 5' to the 3'. Do you see any connection between the mRNA and the 5' to 3' strand of the DNA? If you focus really closely, you're going to find that they are literally equal. They're the same thing. The only difference is that the U is switched with the T. Okay? So the anti-template or the coding strand is the same as the mRNA, with the only difference being that the T is replaced with the U. So in the exam, you need to focus if he gave you the 3' to 5', or if he gives you the 5' to 3'. Remember, 3' to 5' is the template. Okay? This is the one that we'll be using to transcribe an mRNA. And the mRNA that we produce will be in the 5' to 3' end. What is this called? It's called complementary. Complementary meaning that the T will bind to IA. The G will bind to the C. The A will become U. That's called complementary. Complementary. And anti-parallel means that it's in the opposite direction. So this is in the 3' to 5' end. This is the 5' to 3'. Okay? So complementary means the nucleotides are linked together, they complement to each other. And anti-parallel means it's in the opposite direction. Okay? Now, we have an mRNA, correct? 3 nucleotides in the mRNA are called a codon. So 3 nucleotides will form one codon. Why is this codon important? Because this codon, this codon produces a specific amino acid. Now we're going to be talking more about this in translation later on. We're not going to take it in this lecture. But amino acids put together will make a protein. Multiple amino acids will make a protein. So first I had DNA nucleotides, double helix. I unwinded it. I have 2 single strands. The one that I use is the 3' to 5', and it's called the template, non-coding, or antisense strand. From this strand, I will get my mRNA, which is complementary and anti-parallel, basically meaning that it's the A will become U, the C is G, the C is G, and anti-parallel in the opposite direction, which is the 5' to 3' end. 3 nucleotides inside of the mRNA are called a codon, and each codon codes for an amino acid. Okay? I hope this is kind of clear. Okay? Let me just clear. Okay. So a ribosome. What is the ribosome? An mRNA will present the codon to the ribosome. So overall, why are we producing an mRNA? Why do I need an mRNA? So that it can go to the ribosome and start translation. Okay. So first off, I have transcription, which we'll be talking about in this lecture. After it, when we get an mRNA at the end, it will go and it will start translation. Okay? So a ribosome is an intracellular structure made of both RNA and protein. So it's made up of two units, large and small subunit. Inside of each unit, you have RNA and you have protein. And the ribosome, it is the site of protein synthesis in the cell. So RNA translation is the same as protein synthesis. It functions in translation. Okay. So why are we producing mRNA altogether? In order to go to the ribosome and produce a protein. Okay. So this is the end product of transcription, the mRNA. Moving on, the RNA structure. What's the difference between RNA and DNA? First off, the sugar. The sugar in RNA is ribose, whereas in DNA we said it's deoxyribose. Okay. We have a phosphate group, just the same as DNA, on the 5 carbon. We have a nitrogen-containing bases, adenine, guanine, cytosine. However, we have uracil. We don't have thymine like DNA. We have uracil here. Remember this, it is really important, okay? After that, we have a hydroxyl group at carbon number 2. In DNA, we only had a hydrogen. It's single-stranded, DNA is double-stranded, and RNA can store information and catalyze chemical reactions. What does that mean? We'll be talking more about it later on, but remember that the RNA, it can function as DNA, which is storing information, and it can function as a protein, which is catalyzed chemical reaction. Okay? Remember the difference. Remember, this is also a difference between RNA and DNA. Now, I have a question. Which component is not part of the structure of RNA? I'll give you guys 5 seconds to answer. Okay, and the answer is C, deoxyribose sugar. So we said we have ribose sugar, we have a phosphate group, and we have nitrogenous bases. However, deoxyribose sugar is only found in DNA. Okay, remember that. So, types of eukaryotic RNA. Okay, types of eukaryotic RNA. First off, we have the mRNA, which is the product of transcription. Okay, what is the function of mRNA? It transfers DNA code to ribosomes for translation. So in order for the code that's inside of the DNA to leave the nucleus and go to the ribosome in the cytoplasm, we need an mRNA for that. What about the transfer RNA, tRNA? It brings amino acids to ribosomes for protein synthesis. Okay, so we're talking about taking it more in detail in translation. However, I have an mRNA, صح؟ رحت على الريبوزوم. I went to the ribosome. I want to start making protein. I want to start synthesizing protein. Where will the amino acids come from? It will come from the transfer RNA. So the tRNA will bring amino acids to the ribosome in order to produce proteins, okay? Ribosomal RNA, they are ribosomes made of rRNA and proteins. Basically, they are RNA found inside of the ribosome. Heterogeneous nuclear RNA, or hnRNA, they are found only in the nucleus of eukaryotic cells. Remember, prokaryotes, they don't have a nucleus, okay? From the beginning, they don't have a nucleus. So heterogeneous nuclear RNA is only found in eukaryotes. And it represents precursors of mRNA. So before I get an mRNA, I have hnRNA. We'll be focusing more about it later on as well, okay? Just get the concept for now. Small nuclear RNA, it's only found in the nucleus of eukaryotes. One of its major functions is to participate in splicing mRNA, removing introns. So remember, snRNA is for splicing, okay? It's important. I'm underlining the keywords for you guys to remember. Splicing, snRNA, okay? For the micro RNA or miRNA, they control, I don't care about short or long coding, they basically control the expression or repression of other genes during development. So during development, I have, let's say, 10 genes, okay? I have miRNA, it will stop gene number 9, let's say, and it will activate gene number 1 more than the others, Okay, so what type of RNA does this function? It's the microRNA. Okay, ribozymes, they are RNA molecule with enzymatic activity. Okay, so they are basically RNAs that act as enzymes. They are found in both prokaryotes and eukaryotes. Okay, for the example, I don't think you guys are really need to know it, but just know it. Okay, please make sure to know it so that you can guarantee the A in the exam. Peptidyl transferase 23S or RNA. It catalyzes the addition of amino acid residue. Okay, but what you need to really focus on is that ribozyme. It is an RNA molecule with enzymatic activity. Okay, that's what you need to focus on the most. Moving on. Which type of eukaryotic RNA is primarily involved in regulating gene expression? So, what's the key word here? This is how you solve the exam. First off, we said eukaryotic, right? Okay. RNA, regulating gene expression. Okay. So, what type of mRNA? What type of RNA? Sorry, I'll give you guys 5 seconds. So the correct answer is C. If you go back, it's literally written for you in red. mRNA controls the expression or repression of other genes. Okay, these are the small details that you guys need to focus on, the keywords. Okay, moving on. RNA polymerase. What is RNA polymerase? It's the enzyme that controls transcription. What did we say controls translation? Ribosome. So RNA polymerase, it controls transcription. So what does the RNA polymerase do? Step number 1, it searches for initiation site. What is the initiation site? It's the location on the DNA where the ribosome comes and reads it. It will start transcribing. Okay, so I have a region on the DNA. Let's say this region. It's called the initiation site. When the ribosome comes and reads this site right here, it will begin transcribing. Okay. After that, after it begins transcription, it will unwind the double-stranded DNA. So basically, you have double-stranded. It will start unwinding it. So instead of it being like this, it will become like this, just like the picture right here. So it will start unwinding the DNA. Select the correct ribonucleotides, step number 3. Select the correct ribonucleotides. See the ribonucleotides? It will select them based on the DNA and catalyze the formation of phosphodiester bonds. Okay, step number 4. So step number 1, we have the initiation site. It will bind to it and it will start transcription. Number 2, it will unwind the DNA, the double-stranded DNA, making it a single strand. What is this strand right here called? The one that we use for making mRNA, the template strand. Okay. After that, it will select the ribonucleotide triphosphates or the ribonucleotides. It will select them based on the DNA. So let's say I have a DNA with the sequence ATG. It will bring nucleotides with the sequence UGC. Okay, so this is what this step means. Selecting the correct ribonucleotides is basically this step right here. And then it will catalyze the formation of phosphodiester bonds. So when I get these ribonucleotides, I want them to bind to the DNA. So it will form, to bind to each other, sorry. So it will form connection between them. So basically, phosphodiester bonds. Okay. After that, it will detect the termination signal, which is a location also found on the DNA that will tell the ribosome to stop transcription. So basically, initiation site, where the ribosome starts transcribing, termination signal, ribosome stops transcribing. Okay? How many RNA polymerases do we have? We have 3 types, 1, 2, and 3. The most important type, and the one that we'll be talking about most of the time, is called RNA polymerase number 2. Okay? Now, in the exam, it's not going to be written 1 and 2, it's going to be written in Roman numbers. Okay? 1, 2, 3. This is 1, 2, and 3. Okay? So, RNA polymerase number 2, it is responsible for 70 to 80% of gene expression. Okay? And remember, it does not proofread its work. Okay? Just remember this information for later on. RNA polymerase does not proofread its work. It's going to be more important in later on lectures. Okay? Hope it was clear. And then we're going to go into transcription right now. Okay? We're going to begin with transcription. So, first off, we have something called transcription factors. What are transcription factors? They are proteins that bind to DNA near the site of transcription gene. What does that mean? So you have right here, look closely on the picture, okay? Remember, pictures are really important in MOL. Focus on them. Everything in them is really important. So you have the start of transcription. It's this one right here. Before the start of transcription, we have transcription factors that bind to the DNA. Okay? What is their function? They recognize and regulate transcription of different genes. What does that mean? They basically inhibit or assist RNA polymerase. So they either increase the activity of the RNA polymerase or they decrease it. Okay? Now, you are supposed to remember types of transcription factors. So you're supposed to remember transcription factor 2D, transcription factor 2B, transcription factor 2E, and transcription factor 2H. An easy way to remember them, because Dr. Jada said to remember them. Okay, so an easy way to remember them, basically transcription factor 2 bed. And then the 2, if you add a line in between, it will become an H. So these are the transcription factors that you guys need to remember. Okay. The general, okay, so we have two types of transcription factors, general and specific. The general transcription factors, they combine with the RNA polymerase. You see them right here. They combine with the RNA polymerase to form a pre-initiation complex. So pre means before, initiation means beginning. So it will form a complex before the beginning of initiation that is competent to initiate transcription as soon as the nucleotides are available. So basically, they just bind to the RNA polymerase. They either inhibit or assist it. Okay. You're supposed to remember these transcription factors right here. TF2, B, E, D, and H. Okay? And the transcription factors, what do? They bind to the promoter site. So we said they bind to the site on the DNA before the start of transcription. What is this site called? It's called the promoter site. Sorry, promoter site. Okay? So is the promoter site and start of transcription the same place, or are they the same? No. We said that the promoter site is before the start of transcription. Okay, they're not the same. Transcription factors, they bind before the start of transcription. Okay, moving on. We have stages to transcription. We have three main stages. First off, we have the initiation stage, and then we have the synthesis of RNA, then termination stage or termination signal. Okay, so initiation, the RNA polymerase enzyme and DNA form a stable complex at the gene promoter. So we're gonna look at the picture right now. You have the promoter region. We said promoter is where transcription factors will bind to. After that, the RNA polymerase will bind to the DNA. When it reads the transcription unit, sorry, not the transcription unit, when it reads the start point of transcription, it will start transcribing. Okay? After that, you have the synthesis of RNA. So when it reads the start point, when it reads the start of transcription, it will start synthesizing the RNA. So it proceeds using DNA as a template. We talked about it, template strand. So I'm going to use this strand right here. I'm going to call it the template strand. You have the unwounded DNA inside of the polymerase, and you have the RNA transcription taking place. Okay. After that, you have elongation, which is basically just producing more and more RNA, making it longer. After that, you have the termination signal, which is the which basically tells the RNA polymerase, خلاص, stop your work, خلاص خلص شغلك, your work is done, leave. After that, you're gonna have a complete RNA transfer. Okay, so stages of transcription: initiation, synthesis, and termination. Question: during which stage of transcription does RNA polymerase bind to the promoter region? I'll give you guys 5 seconds. It's a really easy question. You should answer this. Exactly. So it basically binds to the initiation. Okay. So in the initiation stage, the RNA polymerase will bind to the DNA. Okay. Moving on. Base numbers, it's a really easy concept. I'll just go over it really quick. The first base, which is the start of transcription, is +1. It's not 0, it's +1. Remember that, okay? To the left of it, to the left means upstream, okay? To the left is called upstream. We're gonna go into the negatives. And to the right, which is basically called downstream, we're gonna go into the positives. So to the left of the starting point for transcription, you have negative, and to the right, you have positives. That's all you guys need to know. And remember, the transcription site, the beginning of the transcription is +1. Now, eukaryotic promoter elements, these are the places where transcription factors bind to, okay? Where transcription factors bind. they lie upstream. So we said upstream means to the right or to the left of the transcription site starting point. Upstream means to the left. Okay. So promoter elements, they lie to the left of the transcription site initiation. What are the promoter elements? Cat box, Kaka box, DC box, cap site. You need to remember these four. So promoter elements are cat, kata, DC, and cap. Transcription factors are transcription factor 2, bed, and H. Okay. So question. Wait, I think the answer is yeah. Okay. So question. During RNA synthesis, the DNA template sequence, TAGC, would be transcribed to produce which of the following RNA sequences? In the exam, you have a paper with you, so make sure to write everything down. TAGC. It does not give us if it's 5' to 3' or 3' to 5'. But always, if it does not give you numbers, expect it to be 5 to 3. Okay? Now, it's 5 to 3, صح? So, is this the template or the anti-template strand? It's the anti-template strand. Okay? So what will I do? I will get, there's two ways to solve this. First off, I'll get the template strand, which is A, T, C, G. This is in the 3' to 5', صح ولا لا? Wait. Wait, never mind, scratch that, okay? I'll just teach you the easy way to solve it. So we said this is the 5' to 3' direction. How did we say the ribosome reads the DNA? We said it reads it in the 3' to 5' direction. Okay. So TAGC, sorry, TAGC will give me A, U, C, G. Why did I do this? Why did I do this? This is the, wait, I always get confused with this. Wallahi, just give me a second. Let me try to. Okay. Okay. I got it. Okay. So TAGC, okay? This is 5' and this is 3'. The ribosome, how will it read the DNA strand in order to make the mRNA? From the 3 to the 5. Okay? So if I'm reading it in this direction, what will I get here? I will get G, C, U, A. 3', 5'. Complementary and anti-parallel. Okay, you see what I'm trying to say? So 3' to 5', I will read it in this direction. I will get G, C, U, A, not T. Remember, RNA is U, not T. And then I will flip it. So basically 5 is here, 5 becomes here. So how will I read this RNA? I will read it from the 5 to the 3'. So if I read it from the 5 to the 3, it will be G, C, U, A. Okay? Do you understand how to solve it? I know I got confused with it. So I probably confused you guys with me. So I'm going to repeat it one last time. TAGC, 5' to 3'. The ribosome, how will it read this sequence? It will read it in this direction. So what will I get? I will get G, C, U, A. And then it's anti-parallel. So I will flip the 5 and the 3. So this is 3', this is 5'. How do I read the RNA? I will read it in the 5' to 3' direction. So I will get G, C, U, A. Okay? I hope it was clear. If you guys have any other question that you don't understand, please send it to me on WhatsApp. I'll be more than happy to answer you because I know this is a really confusing topic. Inshallah, if you have any question, I'll be more than happy to answer it for you guys, okay? Let's move on. Alpha amanitin. I have an entire slide on this, so it's really important to remember it. Alpha amanitin. It's an RNA polymerase 2 inhibitor. So basically, you know, when you go into the desert, they tell you don't eat any mushroom, you see, because some mushrooms have alpha amanitin, and it will inhibit the RNA polymerase 2 inside of your bodies. So it will probably lead to your death. Okay, that's all you need to know. RNA alpha amanitin is RNA polymerase 2 inhibitor. Okay, and it comes from mushrooms. Now, enhancers. Enhancers, they are bases in the DNA that increase the activity of many promoters. From their name, enhancers, they will enhance the activity of the promoters. Okay, they can exert stimulatory actions over long distances, so they can be found thousands of base pairs far from the initiation site. Okay, they could either be downstream or upstream, does not matter. Okay. Now, transcription of several genes on a chromosome. First off, let's look at what promoter is and what terminator is. Okay, so promoter, it will promote the activity of the RNA polymerase. So the RNA polymerase will begin transcribing here. Terminator is the termination signal where the RNA polymerase will leave. Okay, now this is a eukaryote. So in eukaryotes, we have promoters and terminators for each gene. So that's why we have monocystonic RNA. Okay, so why do we have monocystonic RNAs? Because we have a promoter and a terminator for every gene. Okay, what's the difference between eukaryotes and prokaryotes? Okay, in prokaryotes, we have only one promoter and we do not have a termination signal. So what does that mean? When I begin transcribing, I will transcribe all the genes together. So this mRNA contains the genes for all of the proteins. So in bacteria, for example, bacteria is a prokaryote, I have environmental changes and I want to increase the regulation of certain proteins. And let's say gene 1, gene 2, and gene 3 are required for survival. In bacteria, it can produce all of these genes at the same time. Okay, so it does not need to produce gene 1 using an RNA polymerase, and then gene 2 by using another RNA polymerase. One RNA polymerase will transcribe all of these genes, and one ribosome will translate all of these genes. Okay, now Shine-Dalgarno. Shine-Dalgarno is really important, and you guys need to focus on it. Why? Because Shine-Dalgarno is the place. You know what? We'll talk more about it in details later on. But for now, just remember, Shine-Dalgarno. We'll talk about it more later on. But as I said, transcription of prokaryotes can produce many genes inside of a single mRNA. Okay. Whereas in eukaryotes, one RNA polymerase will transcribe one gene at a time. Okay. Some bacterial operons produce polycystronic messages. Poly means more than one. Cystronic means gene messages. So many genetic messages inside of a single mRNA. In these cases, related genes grouped together in the DNA are transcribed as one unit. We talked about it. And they code for several different proteins. I'll just give you an example of how it works. Okay. So, question: what is the primary function of the Shine-Dalgarno sequence in prokaryotic mRNA? I just went over it quick and we'll go more into details later on. But what did I say? 3, 2, 1. It will facilitate the initiation of translation. Okay. So we have transcription, produced mRNA, and then Shine-Dalgarno, it will initiate translation. So as you see here, we have Shine-Dalgarno for each gene. Okay. So when the ribosome, we said ribosome is for translation. When the ribosome comes to the mRNA to read it, it will see the Shine-Dalgarno. It will start translation. It can go and translate these two and stop here. It can translate gene number 1 and gene number 3 without gene number 2. Why? Because each gene has its own Shine-Dalgarno sequence. So let's say I'm a ribosome. Okay, I come to this mRNA. أنا من عندي I want to read this one and read this one. Okay, so I read Shine-Dalgarno number 1. I translated it and produced protein number 1. I did not read Shine-Dalgarno number 2, so I'm not going to translate it. I read number 3, so I will translate gene number 3 into a protein. Do you guys understand this topic, inshallah? Come on. So now we understand Shine-Dalgarno facilitate initiation of translation, not transcription, translation. Okay. Moving on. Eukaryotic transcription units. Okay. So first off, let's go over everything really quick. We said +1 is the start of transcription. So transcription will begin at +1. Upstream from the start of transcription, we have the cat and tata box. What did we say these are? We said they are the promoter elements. And what is the function of the promoter elements? It is the site where transcription factors will bind to. Okay, so transcription factors will bind to the promoter elements and they lie upstream. So they are in the negatives and they are in the promoter region. Okay. After that, we have the ATG or AUG in RNA, and it's basically the start codon. So take a closer look at this. Okay, transcription, it began here. However, the start codon is here. What is this space between these two called? It's called the 5' UTR, or untranslated region. What is the function of the 5' UTR? It gets transcribed but not translated. Function itself, we're not sure of. Okay, we still don't know that. However, remember, 5' UTR will get transcribed. However, because the start codon is after the 5' UTR, the translation will begin here. So the 5' UTR will not get translated. Same thing at this at the 3' region, you have the 3' UTR. It will also get transcribed, but not translated because the stop codon is before it. Okay, just remember that. It's really important information for later on lectures. Let's remember that for now. Okay, so we said translation will start at the start codon and will stop at the stop codon. So I will not have a 5' UTR in my protein or a 3' UTR. Okay, now there is something called the poly-A addition. We'll be focusing on it later on, but remember for now that there is a site at the 3' UTR called the AAU triple A. AAUAA. We'll talk more about it later on. Okay? So I hope this concept is clear for you guys. Now, for the prokaryotic transcription unit, 1 second, okay. For the prokaryotic transcription unit, they do not contain 5' cap or 3' tail. What do these two words mean? We'll talk more about it later on. Okay. So first off, transcription +1. We have 5' UTR, 3' UTR, promoter at the negative, which is upstream. Okay. Everything is the same. صح? Okay. I transcribed it. I formed an mRNA. Inside of the mRNA, you remember the Shine-Dalgarno sequence that we talked about? It's right here. What is the Shine-Dalgarno sequence? Just give me a second. Let me get the charger. Okay, sorry about that. So, what is the difference between the eukaryotic and prokaryotic? The first difference is that there is no 5' cap or 3' tail in the prokaryotic. The other difference is that I have a Shine-Dalgarno sequence. It's basically the ribosomal binding site, as we discussed earlier. So the ribosome will come and bind at the Shine-Dalgarno sequence, and it will start translation. Okay, it will start translation. Where is this found? It's found only in prokaryotes. It's not found in eukaryotes. Okay. Now, the mRNA, it does not have to leave the nucleus, because there is no nucleus in prokaryotes. So in prokaryotes, I don't have a nucleus, so it does not have to leave and then enter the cytoplasm. على طول, transcription and then translation. Okay. It gets translated the minute it's synthesized because it does not have to leave the nucleus into the cytoplasm. And remember something important. Prokaryotes, they do not have introns. Okay, we're going to talk more about it later on as well. But prokaryotes, they don't have introns. So they don't need RNA editing. So the second that I synthesize the RNA, it's ready to be translated. على طول. But in some cases as well, while I'm transcribing the RNA, so basically, this is the polymerase. While I'm transcribing the RNA, it's still going on. I can have a ribosome come and bind to it and start translation. But it can take place in literal seconds, okay? But remember, in prokaryotes, there is no nucleus. The RNA, the second it's transcribed, it can get translated. It does not have to go into further processing. That's the first difference. Second difference, in prokaryotes, I have Schneider-Garner sequences, which are the ribosomal binding site. Third difference, there's no 5' cap. Fourth difference, there's no 3' tail. Okay? So basically, I'm going to ask you a question. Where is the Schneider-Garner sequence? Found in DNA or RNA? The sequence itself is found in both DNA and RNA, because in order for it to be found here, it needs to be transcribed from the DNA. It cannot just, subhanAllah, like appear out of nowhere. Okay? So, I have a question. Which of the following statements accurately describe the difference between eukaryotic and prokaryotic RNA concerning introns and exons? I'll give you guys 5 seconds to answer it. You can pause the video if you want. Yeah? So, the answer is... Okay, the answer is A. So eukaryotic genes, they typically contain introns, while prokaryotic genes do not. I told you, remember, in the eukaryotes, I have introns. That's why I need to further process the RNA that's being produced before I translate it. However, in prokaryotes, I do not need to further change the RNA, so it can get translated out. Okay? Remember that. Now, prokaryotic transcription, really straightforward. You have one RNA polymerase for prokaryotes. In eukaryotes, we have 3. And the one that's most important is number 2. Now, this is the RNA polymerase, okay? For it to actually start transcription, it needs a sigma factor. So sigma is a protein factor required for the initiation of transcription, straightforward. So that I can start transcribing, I need the sigma factor. When I start transcribing, when transcription takes place and starts happening, the sigma factor is released. Okay, so I do not need a sigma factor when I start transcription. Okay, however, I needed to start it. Okay, I'll go over it one more time. So to start transcription, I need sigma factor. When I already started and transcription is taking place, it will be released. I don't need it anymore. Okay, so as you can see here, transcription is taking place. I don't need it anymore. How does the RNA polymerase know when to stop? We have something called RHO factor. The Rho factor is a protein factor required for termination. So when the Rho factor comes and binds to the RNA polymerase will leave and it will stop transcription. So for initiation, I need sigma. For termination, I need Rho factor. Another important thing, we said alpha amanitin, it will inhibit RNA polymerase 2 in humans, in eukaryotes. In prokaryotes, we have something called rifampin. Prokaryotic RNA polymerase, it's inhibited by rifampin, which is a type of like drug that we give to patients with tuberculosis or TB. You don't need to know in more details, just know that rifampin, it can detect RNA polymerase for prokaryotes. It does not detect RNA polymerase for eukaryotes. That's why it's an effective drug. It will not kill us, however, it will kill the bacteria. Okay? Now, comparison of RNA polymerases. In prokaryotes, it's a single RNA polymerase. In eukaryotes, I have 3 types. RNA polymerase 1, it's found in rRNA. RNA polymerase 2 is for hnRNA and some snRNA. Number 3 is for tRNAs. Do you need to remember these? Yes, let's remember them to be on the safe side, okay? Now, prokaryotes, they require sigma to initiate. Eukaryotes, they need transcription factors bind to the RNA polymerase in order for it to start, okay? And prokaryotes requires RHO factor. Eukaryotes does not require. Prokaryotic is inhibited by rifampin. Eukaryotic inhibited by alpha amanitin, okay? We're done with RNA transcription number 1. We're gonna go into number 2. Number 2 is the more fun RNA transcription lecture, okay? So, RNA processing. It is the process through which the primary RNA is further processed. What did I tell you? The RNA we produce in eukaryotes need to be further processed in order to be translated. In prokaryotes, the mRNA that's transcribed gets translated outward. Okay? Remember that. What are the steps in RNA processing? Number 1, we have the addition of a cap at the 5' end. We said that prokaryotes do not have it. Number 2, we have the addition of a poly-A tail at the 3' end. Remember, we said there is no 3' tail in prokaryotes because we do not need any further processing of the RNA. Do you see how you connect the dots? We have RNA processing in eukaryotes not in prokaryotes. So that's why I don't have these two in prokaryotes. After that, we have RNA editing and RNA splicing. Okay? So let's go one by one. First off, we'll be talking about the cap, the 5' cap, and the poly A tail, which is what, sorry, this one right here, and the poly A tail. Okay? So first off, we talked about hnRNA. We said that hnRNA comes before the mRNA. So you see all of these processes right here. RNA processing is hnRNA. The product of RNA processing is mRNA. Okay? See how you connect the dots between the two lectures? So first off, we're going to talk about the cap, 5' cap, and the poly A tail. 5' capping, we have, let me just clear the drawings. We have a 7-methylguanosine cap. 7-methylguanosine cap. Remember the name, okay? 7-methylguanosine cap is added to the 5' end of the RNA molecule during its synthesis. So 5' end, I have a cap. What is it called? 7-methylguanosine cap. What is the function of it? It's a site for ribosome binding. Site for ribosome binding. So the ribosome comes to it and it will bind to it. And it's a protective feature that prevents the mRNA chain from degradation. So basically, it will make it more, what's it called? More stable, okay? So if I do not have a cap, the mRNA is more prone to being degraded. Okay? Two functions of the 5' cap. The poly A tail. The poly A tail is added to the 3' end of the RNA molecule. So at the 5' we have cap, 3' we have tail. Where is the poly A tail added to? This process begins when an endonuclease cleaves the RNA just downstream of the AAU-UA. Okay, so on the DNA, I have a sequence that's AAU triple A, U triple A. When I read this sequence, that's where I will cleave the DNA and I will stop transcription. So the end of transcription will be right here. The following this, poly A polymerase, remember the name, poly A polymerase will attach a tail of approximately 200 adenine colony. Okay. So what's the poly A? It's multiple. Poly means multiple. A. So I have a lot of A's back to back. A. The poly A polymerase will bring these this poly A tail and attach it to the AAU triple A. Okay. Now, what is the function of the poly A tail? It helps safeguard the mRNA from rapid degradation and facilitate its transport to the cytoplasm. So it will make it more stable, and it will bring it from the nucleus into the cytoplasm. So we said we have 5' capping. Its function is ribosome binding site and makes it more stable. Poly A tail, it will bind to the 3' region after the AAU triple A, and it will make it more safe, sorry, not more safe, it will make it more stable, and it will transport it to the cytoplasm, okay? After this, we have the splicing of the mRNA by spliceosomes. Remember when I told you to memorize the names of the RNAs? We said spliceosome contains snRNA, okay? What is their function? They remove the introns. You see the introns, the non-coding part of the mRNA, the spliceosome will come and it will remove it. Now, let's say I have exon, intron, exon, intron, okay? The spliceosome will come and it will cut this intron, and it will cut this intron right here. Then they will join these introns together, and then it will form something called lariat, okay? So all of the introns that I cut out will join together and it will form a lariat, which is a structure that will get degraded in the nucleus. So exons with introns. I remove the introns. These introns will be degraded in the nucleus. I don't need them anymore. I will get rid of them. Okay? Let's see it in text. So intron splicing. Introns are removed from the heterogeneous nuclear RNA through a process called splicing. Intron splicing, which is carried by spliceosomes, also known as small nuclear ribonucleoproteins, SNRLPs. Okay. These complexes consist of small nuclear RNAs and proteins. We talked about it earlier on. During the splicing, the hnRNA is cleaved at the splice site located at the 5' and the 3' ends of the intron, also called donor and acceptor. Do you need to remember the names? Yes, you do. Okay. So on the intron, at the 5' end, it's called the donor site. And the 3' end is called the acceptor site. How to remember it? Basically, I cut it right here, صح? But exon will donate its binding to the exon on the other side, which is called the acceptor. Okay, that's just an easy way to remember it. That's how I remember it. Okay, so the 5' of the intron is called the donor. The 3' is called the acceptor. Okay. The intron is excised in a lariat structure and subsequently degraded. We talked about it. Okay. After the introns are removed, the exons are joined together. Now we have something called alternative splicing, and this is a really important topic that you guys need to focus. In certain genes, the initial RNA transcript can be spliced in multiple ways. Alternatives, or alternative means more than one way. So the RNA can be spliced in multiple ways, resulting in the production of different proteins. What does that mean? Basically, I have the mRNA right here, okay? mRNA. Alternative splicing. Let's say I decided to splice this one right here. I don't want exon number 3. I want only 1, 2, and 4. I will get protein number 1. However, I don't want protein number 2 this time. Okay? أنا بدي 1, 3, or 4. I will get protein number 2. These two proteins have completely different functions. They could have the same function, but they could have completely different functions. So from the same RNA, I have many different proteins that can come out. So that's why we have around 22,000 genes in our body. However, we have hundreds of thousands of proteins. So how is this accomplished? Through alternative splicing. Okay, so I have a question for you. What is the primary benefit of alternative splicing in eukaryotic cells? Pause the video if you want to answer it by yourself. But the correct answer is B. It allows a single gene. Let me go back. It allows a single gene to produce multiple protein isoforms. Okay? So a single gene can give me multiple proteins. That's called alternative splicing. Now, he gave you two examples that you guys need to remember. The first one is the variants of the muscle proteins, tropomyosin and troponin T. Okay? Tropomyosins and troponin T are produced in this way. They all come from a mother mRNA. Okay, so this right here, this is the mother mRNA. Alpha tropomyosin gene is organized into 15 exons on chromosome 15. So I have 15 exons in the alpha tropomyosin gene, TPM1. Let's say أنا بالأول I decided to splice it like this. Hello? I will get striated muscle mRNA. The second time I decided to splice it in this way. I will get smooth muscle mRNA. المرة الثالثة يلا I will splice it this way. What will I get? Fibroblast mRNA. المرة الرابعة I decided يلا let me splice it this way. I will also get fibroblast mRNA. The fifth time I decided let me splice it this way. I will get brain mRNA. So you see that the single difference in splicing, can form a completely different product. If you take a closer look right here, this is the mother mRNA, and all of these are products from a single mRNA. Subhanallah. Okay, we have 22,000 genes in our body that code for hundreds and thousands of proteins. Okay, how does that happen? Through alternative splicing. And we just took one example. Now we're going to take the other example. IgM and IgD. IgM and IgD. You guys took it in foundation in the HLS part. These are antibodies. Okay, how are they generated? By alternative splicing. I'm not going to go into details about this. We just need to get the general idea. You have the mother mRNA right here. Okay, mother mRNA. I have a stop codon right here, and I have a stop codon right here. أنا جيت حبيت I am the RNA polymerase. The first time I decided to ignore this stop codon and only read this one right here. What will I get? I will get, wait, this IgM or IgD? Yeah, I think this is IgM. Okay. So the longer one is IgM. I decided to remove this one and only keep the other one. So I will get IgM. In the other case, I decided to keep this one and remove this one. What did I get? IgD. So just get the general idea, okay? Basically, IgM and IgD, they share the same mother mRNA. The difference in splicing, which is called alternative splicing, will result in two different antibodies. Okay, that's all I want you guys to remember. Now we're gonna go into diseases. The first disease is called beta thalassemia, or not disease, sorry, mutation. So in alternative splicing, while I'm splicing, I could have mutations, and this can lead to abnormal protein formation. So example of this is beta thalassemia. Mutations that disrupt the correct splicing of beta globulin mRNA can lead to certain diseases, that cases of beta thalassemia, a condition characterized by reduced or absent production of the beta globulin gene. So I have the normal beta globulin gene right here. Okay, I have a mutation in splicing. بالغلط, by mistake, I kept all of this exon right here. This will result in beta thalassemia. Let's say I only kept a portion of this. It's still a mutation, but it will result in beta thalassemia. So a mutation in splicing can lead to diseases. Okay, in this case, beta thalassemia, because I have abnormal beta globulin genes. Okay. Another example, it's systemic lupus erythematosus, which is called SLE. SLE is a chronic autoimmune disease that affects almost any organ in the system, or any organ system. How does it occur? In my body, I do not have antibodies against FNRP. I do not have them. But by mistake, I have produced a class of antibodies that is against spliceosome. Okay, what will happen? I will have defects in my mRNA splicing. Okay, spliceosome, from its name, splice, they are the ones that are responsible for the splicing of the mRNA. We just talked about it. So if I have an antibody produced against the spliceosome, I will not have functional SNRP. I will not have functional splicing. I will have a disease called SLE. Okay. Another example of a disease that could come from mutation in splicing, Hutchinson-Gilford progeria syndrome. Okay, HGPS. All I need you guys to remember is that HGPS is characterized by fast aging in children. So basically, a child that's 13 years old looks like a person that's 70 years old. Okay. Why is this caused? It's caused by the mutation in the LMNA gene. That's all I want you guys to remember. HGPS comes from the mutation in LMNA gene, and it's characterized by the aging, the fast aging of children. Okay. So I have a question. RNA editing refers to any type of modification in the RNA transcript sequence that occurs without involving. Sorry, it's not a question. Okay. RNA editing refers to any type of modification in the RNA transcript without involving RNA splicing. So we said RNA splicing, it involves cutting introns. صح? I remove the introns. RNA editing, which is step number 3, it occurs before splicing, and it's any modification, addition, deletion, anything that does not involve splicing. Okay, what do I mean by this? It allows for, first off, this RNA editing allows for greater protein diversity. RNA editing involves altering the RNA sequence through modification, insertion, or deletion of one or more nucleotide bases. So I'm not splicing them, I'm simply removing, adding, or changing. Now, we have 3 types of RNA editing reactions. The first one, substitution of uridine with 4-cytidine. What does uridine 4-cytidine mean? Basically, 1 second. Okay, so the U will replace the C. So if in my sequence I have CAB, sorry, خلينا على نفس المنطقة. Okay, CAG, the C will be replaced by the U. Okay, that's what substitution of uridine for cysteine. The cysteine will be removed and will be replaced by a U. Okay, and easy way to remember this is see you later. C U. Okay, the C will be removed and the U will take its place. The other one is substitution of inosine for an adenosine. So I have, let's say on the same example, CAG, it will become CIG. So the A is replaced by an I. AI, artificial intelligence. A will remove and the I will take its place. Same with the CU. The C will be removed, the U will take its place. Okay? The third one, the third type of RNA editing is called guide RNA or gRNA. Basically, it is site-specific insertion, deletion of uridine base. All you need to remember, okay? Guide RNA, insertion and deletion of uridine base. That's all you need to remember. Now, for the number 1 and number 2, we'll talk about it in more details now. Now, C to U substitution is carried out by cytidine deaminase enzyme. So, what will bring the U and replace it? What will replace the C with a U? Cytidine deaminase enzyme. It will convert the cytidine base into a uridine base. Okay. However, A to I substitution, it involves an enzyme called adenosine deaminase acting on RNA. Why is it important to remember the acting on RNA part? Because this enzyme is different from the adenosine deaminase involved in purine nucleotide salvage. So C to U, it's done by an enzyme called cytidine deaminase. A to I is done by an ADAR, adenosine deaminase, acting on RNA. Not adenosine deaminase, ADAR. Okay, remember this, it's a trick question in the exam. A to I is the most frequent type of RNA editing. Why? Because the inosine, looks like a guanosine. Okay, it gets transcribed and translated easily. That's all you need to remember. Okay, so cytidine, uridine, they get replaced because I'm simply just replacing this group right here. Inosine and adenosine, I'm simply replacing this group right here. Okay, you see the difference between the two? And because inosine and guanosine look the same, they get transcribed and translated easily. So A to I deamination is the most frequent type of RNA editing. Okay? Inshallah, it was clear for you guys. I will go into examples now, since I don't want to take a lot of time. So, example of C2U deamination. Cytidine deamination occurs in tissue specific. So basically in the intestines, I have cytidine deamination. For example, بالضبط هي example. In the intestines, cytidine deaminase will catalyze the deamination of cytosine to mRNA of the ApoB100 gene. What do I mean by this? So, I have normal transcription and translation. I will get ApoB100. However, if I have an RNA editing process, right here, you see CAA, RNA editing will replace the C with a U. So what will happen? I will have ApoB48 and ApoB100. So this modification will produce two distinct proteins, ApoB100 and ApoB48. Okay? I think that's done. Yeah. If you guys have any questions, since this is a recorded session, I went over it a bit, trying to go over it a bit quicker. But if you guys have any questions, please feel free to reach out at any time. I'm more than happy to help you guys. I know that the midterm is coming up, you guys are stressed, but please, if you have any questions, feel free to reach out, okay? Please make sure to scan this QR code for the feedback. And yeah, thank you. If you guys have any questions again, feel free to reach out. I'm more than happy to answer you guys' question. And we're done with this session. Okay. Take care, everyone. Inshallah, you guys have a nice day.